Doesn't look like your audio is joined at all now. Michelle, can you hear me? Might have to call in with the phone.
Hi, Hugh.
Yes. I can hear you.
Yeah, I mean, I usually use my phone because I always get difficulties with the computer, but now it has audio. I have a headset. It still doesn't seem to be working.
Yeah, sometimes it's just a matter of assigning that headset to this meeting. Like when you join WebEx, you have to tell it what you
right.
need to use.
So now I'm hearing. Now I'm hearing echo.
How about now?
So I'm gonna take it off and let me see what's happening. Can you hear me?
Yeah, are you still hearing echo?
No, I took off the headphone.
I also muted the computer so the audio won't go through there.
Yeah. I really have no idea.
About what?
Yeah, I'm still hearing an echo. So it's picking up my voice somewhere. Maybe it has an inbuilt recorder, microphone on the desktop. Okay, it stopped now. Yeah, one of me is gonna leave. Hopefully I get the right one. Yeah. So that's how I can't see who's joined the meeting because
If the echo went away, why did you drop out of the meeting from your computer?
Because I'm using it from my phone.
But now you can't see, you can't see anything in the meeting, or you're in the meeting.
I don't really have to see the meeting. It would be nice to see who's joined the meeting. I don't know. Is there any way to record who's joined the meeting?
Record in like track attendance.
Yeah.
It would be in the participant list. Typically we don't like curate that stuff.
Okay. I just have a quick question about before Dr. Tang joins. So the PIs, they want to use one WebEx for the entire day, right? And I just wanted to find out if there was any issue with the recording, if it's from 12 to 1:30. Does it really matter? We can just record from that time because the WebEx would be from like 9 o'clock until 3. Or does it have to be a specific time?
Let me make sure I'm understanding your question. You're talking about this meeting right now that's going to go from 12 to 1:30 with this WebEx that I'm.
Right. So we only want
I just started the recorder just now.
Yeah, we only want the period from 12 to 1:30 to be recorded.
Right. Yeah.
Okay.
Dr. Tang has joined.
Yep.
Hello.
Okay.
Hi.
Can hear you. How are you? This is Hugh with AV.
Oh, great, thank you. I hear an echo.
Hi, Tang.
Hi.
Do you hear an echo at me too, Hugh?
Nope, I don't hear any echoes.
No,
Yeah.
I don't.
I'm hearing a little bit of an echo as well.
Hmm.
Turn my on.
Shall I try to share my screen?
Yes, please.
Okay, yeah, now the echo is gone, so everything is, so let's try, let me see, so there's, I guess how's that? You can see I should put in the presenter mode. Do you still see the right screen, or?
Yep, it looks great.
Now the echo is back. Oh, it's gone again.
I think it's actually when Hugh is speaking. I think it's just background noise when you unmute and speak.
Hmm. Oh, here's my access control. Okay. This is good. It's similar to Zoom. No huge surprises. Let me put this somewhere else. Can you see my arrows if I point? Or maybe just... Ooh. Okay. Oh, here. Cool.
And probably later today, I'll shoot you an email with kind of some of the stuff that we were talking about, and more clarity about what I was hoping to do. You can kind of give me your thoughts.
Yeah, that will be fun.
Super helpful.
Yeah.
Be back in the same sandbox again.
That'll be fun. I've been taking some notes along the way so that I won't forget, because things just start to pile up.
I hope you had some good meetings, and
It's been fantastic.
Good.
It's a lot of fun. That's good. I was also thinking that I don't know if it's possible, I guess, from the visitor perspective, is it possible just to have one room that people come and visit?
We actually started before you got on the call. We were talking about that because all the PIs have requested that. Some of it is just from a recording perspective. We're going to look into it.
Yeah, the seminar is going to be, I think, it's okay, right? And so that way, I guess it's more seamless. In terms of people can knock on the door to come in, and you realize that you are not going over time. Yeah. Others, who knows?
Yeah. I hope people were gracious about.
No, well, good. It's my fault. I keep on talking and then didn't realize time. There was also a link that was, I think I was going by itinerary. It was linked to a different meeting, and then I was trying to find the right meeting room. But
that's good. That's good feedback, though. We'll try to coordinate that, because that's what we usually do in person as well. Is just have you in one room, and just so you're not walking around so much. So.
Yeah, in person, at UCSF we do get people to escort it to different rooms. I figured that the speaker may appreciate getting up and move around a little bit. Now I'm anchored, chained to my chair. Might as well just, it's not a big deal. It's been good, fantastic. I'm glad that made it this time. Last January will be more fun to visit. It just didn't happen.
Yeah.
Yeah.
Your focus aligns so well, I think, with our group. I thought it would be so exciting, and then with the myeloid stuff that I've been working on, I figured it'd be a nice time to reach out again.
Yeah, we'll see. I'm not the myeloid person, but we'll see. Because it touched on Tregs, and I figured we better study it. And originally, the hypothesis was that Tregs shut down the myeloid cells. I think in the end, the answer is likely it's indirect. It eventually shut down the myeloid cells, but it's because of myeloid cell turnover. With change of the environment, you have a different kind of myeloid cells in the tissue.
Yeah, that's been my hypothesis as well, is that it's more of a production regulated event on the myeloid side, or tissue recruitment, but it's less about kind of a distinct subset.
Right, and then they also change. I found the myeloid cells are challenging to study. it could change even to people image in situ, right? In PAR cubes imaged in situ in the liver, they they flip between lysis-C high and the low.
Yeah, the markers start to get dizzying,
Yeah,
for sure.
yeah.
Some of the work out of, iStar have been really impressive recently with the single cell RNA-seq. And one of the guys I was in graduate school with, Lai Guan, is really pushing that idea that neutrophils and some of the monocyte-derived populations, and what are those precursors and what regulates those. So I think we'll start to know more now that we have better tools.
Yeah, and my early realization of this whole thing was the immediate benefit is I don't just read the paper. I don't search paper for dendritic cell anymore. Because I realized that for the same identical cell population, some people call it dendritic cells, some people call it the macrophages.
Yeah.
It's all the CD11C positive cells in the islets.
That's right. So I think, I hope it was clear, the format, we really gave an hour and then a half hour for questions.
Oh, okay. That's very generous. Yeah, I saw that. There's a half hour. Oh, shoot.
So,
okay.
Hi, Tang.
Hi. I don't know who's talking because I'm
It's
after
Jane.
sharing the screen. Oh, hi. Cannot tell this. How do I get this to be bigger so I can see who's talking?
if you click on layout.
Okay, now it's here. It doesn't show everybody. I guess, oh.
I turned off my video, sorry.
Oh, okay. All right. I'm
wanted
glad you're here. Okay, that's great. Yeah. Yeah.
Hello, hey
All
Tang.
right. Oh, hi, David.
How are you?
Is that you? Good, good.
Thank
The meeting was your lab. I think we have another meeting later, right?
Yeah, thank you for doing this. Really appreciate it.
This is great. I had a lot of fun already.
So maybe we'll give a few more minutes and then we'll get started.
Yeah. So how related physically or as a community of the Benaroya and Seattle Children's and then in UW?
So Benaroya is up the hill. We're both downtown. So the Seattle Children's Research Institute's downtown, and so is Benaroya. So we're actually not too far from each other. And University of Washington has a South Lake Union campus that's downtown as well. So those three are not too far from each other. The main University of Washington campus is a little bit farther away. And then the main children's hospital is also over kind of a body of water. So those are a little bit farther away.
Pretty much all of immunology is focused in this sort of South Lake Union town area now. So it's really a nice cluster of different groups.
I'm just going to say that Seattle Children's comes to BRI to drink our coffee.
That close, okay.
Not anymore.
When we have joint lab meetings.
Oh, wow, that's fantastic. So it's actually quite closely knit community. Yeah. seminars, and you all go to each other's seminars. And
Each of the institutions has their own seminars, and we cross into the different seminars. It's actually great immunology from basic to a lot of translational work. It's really
Yeah, Jane, that's another downside of COVID.
I know we don't get to have our lab meetings. We also provide treats to entice them up the hill.
Okay, so why don't we get started? Do you guys agree?
it's a good time to go.
Okay. So I'd like to extend, together with everybody here, a warm welcome to Dr. Qizhi Tang. Tang has an impressive track record and collection of work that spans a long time on regulatory T cells, specifically with type 1 diabetes, as well as more recently some novel therapeutics for diabetes. She originally started training in Peking Medical College and then switched gears from a medical degree to a graduate career, went to University of Illinois in Chicago, and then did her postdoc with Jeff Bluestone, where we interacted. I met her at University of Chicago, and that's where it was super exciting rediscovering kind of regulatory T cells. And with all that work, she when Jeff Bluestone got recruited to UCSF, she also was recruited and has been doing amazing work. It's kind of now balanced 75% on engineered regulatory cells and 25% on islet transplantation as possible mechanisms of novel therapeutics. So we're super excited. We were able to snag you back to reschedule, and excited to hear your talk.
Thank you so much, Eric. And it's fantastic virtual visit. I wish it was real. It was just January. It was didn't happen. I really sorry that I have to reschedule until this time, but it's been fantastic. So I guess for today's talk, I'm going to focus on Treg therapies and then and make an argument that antigen-specific Treg therapy will be better, and then and try to see if there's a way to develop that therapy for clinical translation. And just to start, and here are my disclosures. I have some patents on Treg cell therapy, and I had funding previously from Juno Therapeutics to develop an engineered Treg therapy program. And some of you may know that I, together with Jeff and Bluestone and Fred Ramsdell and Sasha Rudensky, we funded Sonoma Biotherapeutics for developing Treg cell therapy. So just as an introduction to bring everybody onto the same page, and is that we know that the immunological health is a balancing act. And I guess nothing better illustrate this than COVID-19, right? And you need to mount the immune response to control infection. But when the immune response cannot be brought back and then suppressed once the pathogen is controlled, and then you have uncontrolled inflammation, and that could be pathogenic as well. So you need to balance the activation and also the suppression. There are multiple mechanisms that bring immune system back to balance, and in this the suppressive mechanism include regulatory T cells, regulatory B cells, regulatory dendritic cells, macrophages, NK cells. I think, and it seems that every cell type has its own regulatory pathway. But we focus on Tregs, and it's because it is a specific designated lineage of cells for this task, right? And then it has its own transcription factors, and then it has its own development pathway. And in their absence, and you all know very well that an IPEX patient, and then they develop overwhelming inflammation, uncontrolled immune response. And so Tregs, this is an old review. And then I think one of the reason Tregs are so potent in controlling immune response is because they're very versatile. There are many different mechanisms that they can suppress immune response in different stage of the immune response, right? Just this morning, I was talking to Steve, and then there are yet new suppressive mechanism, but Tregs are being discovered. But in general, the way we think about this is Treg control immune homeostasis. So in the homeostasis and in the absence of onset of immune response, there's a set of mechanisms that control the immune response. When you actually mount an immune response into infection, then Treg actually escalate their suppressive function, then acquire more suppressive function to bring it back to control. And then one important feature of regulatory T cells is this concept of infectious tolerance. By maintaining immunoquiescence and an antigen exposure of other cells in the immunoquiescent stage, and these cells then learned to have regulatory function and become these what we call adaptive or induced regulatory T cells. So in this way, the Tregs not only dominate over other cells, but also dominate their effect over time. So we think it's very hard to recapitulate all these activities using small molecules or biologics. And so in fact, maybe Tregs themselves is the best therapeutics. So I'm going to go into the data presentation. It's mostly on 3 parts. It's initial preclinical data that convinced us to launch a Treg cell therapy program at UCSF, and then and then go back from that clinical experience, and then go back to the bench on what we are doing to improve regulatory T cell therapy, hopefully. So in the preclinical piece, I'm going to focus on the topic of Treg specificity and their mechanisms of action in vivo. Okay. So we started this way back in early 2000 is when we transferred a BDC 2.5 Tregs. This is islet antigen specific Tregs in the NOD mice, and initially into the NOD CD28 knockout mice. And these mice have a deficiency in Tregs. And so they develop a very early and then onset of diabetes in all nearly 100% of the mice. By giving a single infusion of regulatory T cells from these BDC 2.5 TCR transgenic mice, you prevent diabetes completely. And as few as 50,000 cells can achieve this kind of therapeutic effect. And then later on, we also use the BDC 2.5 Tregs to treat new onset diabetic patients, and then could achieve disease remission. What's when we compare to polyclonal Treg, is I'm not showing the data here, but if we use just not Tregs, polyclonal Tregs, 5 million cells will not achieve much of a protective effect. This tells us that the antigen-specific Tregs in the type 1 diabetes model, it's more than 100 times more potent than polyclonal Tregs. Another feature that we realized is the specificity of this effect. So as I mentioned, the BDC 2.5 Tregs are specific for islet antigen. And then the mice actually survived. They never developed diabetes, but by 20 to 25 weeks of age, they become sick, and they eventually died. And when we analyzed these mice, we found this striking phenomenon that the exocrine pancreas disappeared. So this is one of the histology slides from one of these Treg rescued mice, and a not diabetic, actually hypoglycemic at the time when the mouse died. And then pancreas actually turned into a spider web. You cannot even see the exocrine pancreas like you've seen here is a normal pancreas look like. But they're beautiful large islets floating on this web, of what previous to be pancreas. So these mice, in the end, we published a paper on here. They develop exocrine pancreatitis as autoimmune CD4 mediated disease. The BDC 2.5 Tregs specific for islet antigen can control the islet autoimmunity, but not the neighboring tissue exocrine pancreas, and it shows a high specificity of the control. And so how do these cells work? And then the first feature I would like to bring to your attention is that these cells are inflammation seeking. So this is a immunofluorescence staining zoomed in view of a inflamed islets. I'm sorry, I forgot to provide the legend. The red is CD4 and the blue is FOXP3. And the green is the 511 marker that's on the transfer Tregs. And the hole in the middle here is actually the remaining islet mass. It's if I stain for insulin, it will be positive for insulin. But this islet is heavily infiltrated. You see that the Tregs actually come to this inflamed islets, and they actually tend to be enriched at the border between the healthy islet and the infiltrate. And then this border has a lot of dendritic cells that we think that is what the Tregs are interacting with. If you look at these islets and with different degree of inflammation, right, we call it insulitis and mild, moderate to severe. The more severe the inflammation, the more Tregs get in. So they have an intrinsic ability to respond to inflammation and concentrate their effort in the tissue. Another feature of this in this model is the speed of the response, the protection. This is an RNA expression analysis looking at insulin 1 and insulin 2 message. Mouse have two insulin genes. And this is at the time, I think about 6 weeks of age for the not CD28 knockout mice. The disease is highly synchronous. So this allowed us to do this kind of analysis. So you can look at, we normalize to the insulin 1 and 2 expression to 1 is a time of treatment, right? And so 3 days after the treatment, and you see the insulin 2 message start to decrease, and this is a progressive decrease of insulin 2 message, but with Treg treatment, that level is stabilized, and it's similar to insulin 1. In a matter of few days, and then the islet cell mass loss has been stopped by the Treg treatment. So we look at other genes.
In that experiment, the T effector, the antigen-specific T effectors were transferred also with?
No, it's not. Sorry, it's in the CD28 knockout mice. So
28.
this is we take the mice at about 6 weeks of age, 5 to 6 weeks of age, and then transfer and then take out. So these are not the same mice, right? Obviously. So this is a 5-week-old, 6-week-old CD28 knockout mice, and then here is then some of the mice were treated and some were not treated, and then 3 days later after the treatment, we take out these mice and analyze their insulin production in the islets. Does that make sense?
Yes, thanks.
Okay, yeah, sorry. So this is the same kind of experiment, and it's only showing day 7, but we also have the day 3 data is similar, just lower, a different magnitude of the response. To look at how does Treg therapy change the inflammation. And this is by a qPCR analysis of a panel of 90-some genes. And this is it on here. And this is comparing the gene expression in Treg treated versus age-matched non-treated mice. And then you can see here that the genes that are downregulated by Treg treatment and have this Th1 and CTL signature, gamma interferon, CXCR3, granzyme A, B, all right, and CXCL9. So it looks that within days, the Tregs shut down a Th1 and CD8 response within the islets. What I'm not showing here, I think, Eric, you might be interested, is that original hypothesis, we thought, because by 2-photon imaging, we saw Tregs interacting with dendritic cells in the islet. We thought they were shut down the dendritic cells and preventing further T cell activation. That's proven not to be the case because the dendritic cell phenotype change took much longer, at least 2 weeks to manifest. So we think it's a consequence of controlling the inflammation here that led to the change of the dendritic cell phenotype instead of dendritic cell change drove the suppression of the immune response. So just quickly, how does the CD8 and then Th1 response, how Tregs control the response? And then here we did adoptive transfer experiments in these mice. We give Treg cell therapy and also give a 8.3. This is the islet antigen specific CD8 TCR transgenic T cell and BTC 2.5 conventional cells, and then looked at their proliferation in the pancreatic lymph node and versus the islet. You can see here in the pancreatic lymph node, you see this with Treg therapy, there's control of expansion, clonal expansion, proliferation of these islet antigen specific cells. But that's not the case in the islets. We think it likely is because the potency of the antigen presentation in the islets and also the amount of antigen is much higher in the islet. So the Treg did not control their proliferation at all. But they shut down the gamma interferon production. And here is looking at the protein level. So to do this, the top is adopted transfer CD8 and BDC 2.5 cells. And then the bottom is endogenous CD4 CD8. To really look at gamma interferon protein expression inside the islets, what we did was we took these mice, actually injected bufalin A into the mice and then let them sit in the mice for a while and then take the cells out, take the islet out, and then measure the intracellular gamma interferon production. This is the percentage that you can detect. And it's drastically shut down, nearly completely wiped out in the adopted transfer cells and then also in the endogenous cells. And then much to our surprise, and this is what I discussed with Steve earlier today, is when we look at the gamma interferon message on these cells, the CD8 and CD4 cells, we isolated out of the islets. And the message remained similar and then was not shut down at the message level, but we think it's shut down at the translational level. And then we trying to connect the Treg therapy and then trying to understand how Treg could actually shut down the translation inside the effector cells. And we think at the time that it must be through IL-2 or cytokine deprivation. To simulate the cytokine deprivation effect, what we did was took these infiltrating cells and then culture them in vitro and measure gamma interferon production. In some of the cells, in the split of the cells, we actually treated with a JAK3 inhibitor to stimulate a stop of IL-2 signaling. And you can see the continued gamma interferon production depend on JAK3 activity. And so when you inhibit JAK3, you completely inhibited phospho S6 level, as shown in both CD4 and CD8, and it's more dramatic in the CD8 than in the CD4. So this gives us a marker to see if this is indeed the effect of the Tregs in vivo. So we went back into the islets and on histology sections and stained for CD8 in red and then phospho S6 in green. You can see in these CD8s, you can readily detect the CD8 positive and phospho S6 positive cells in the untreated animal, but in the BDC 2.5 treated animal, and then the phospho S6 was completely shut down in the CD8 cells. We still see some phospho S6, and these are actually not in immune cells at all. It's likely in the pancreatic islet cells, beta cells. So we think what Treg does in this model of type 1 diabetes is they infiltrate the islets. I didn't show you that actually they burst into proliferation within the first 3 days. And then they likely through IL-2 consumption and then shut down the effector CD8 program. And that bring the down the inflammation, allow the tissue to gradually turn over the inflammatory cells, including myeloid cells, and then maintain immune quiescence that way. And then this is when I was in the diabetes center, and then when I got my job in transplantation, I decided to try cell therapy in transplantation. I decided to work on islet transplantation for my interest in type 1 diabetes. And then this is our initial experiment. You transplant islet. This is biopsy into B6 animal, and they reject in 2 weeks. And then we give Tregs, we give antigen-specific Tregs to the islet. We give polyclonal Tregs, and no matter, we give 5 million, 10 million, 25 million, and no matter how many we give, I think the most we have given was 50 million Tregs. That did not change the rejection course much at all. And then this got us to think, what might be the difference between a autoimmune disease versus a transplant rejection. And then we suspect it's because of the precursor frequency that can reject a transplant. It's to an alloantigen. It's much higher. It hasn't gone through thymic selection. It's not deleted and not weakened by thymic selection, right? So we figured we must delete some of these donor-specific cells and to give Tregs a therapeutic window so they can control the rejection. They are not meant to control all form of the immune response. So to delete donor-specific cells, what we did was to give donor-specific transfusion and then wait for 2 days, and to drive, the idea is to drive the donor-specific cells into cell cycle and then give them cyclophosphamide. And so that to preferentially kill these activated and proliferating cells and to cause donor-specific deletion. And then we make the mice diabetic and transplant. So if you just do this alone with donor-specific T-cell deletion, you have some prolongation of the islet graft, but not by far. But if you now combine this deletion with the donor-specific Tregs, and here we transferred 5 million of them, now you can achieve long-term graft survival in majority of the mice. So when we do polyclonal Tregs in this setting, at the same dose, 5 million, we definitely achieved a prolongation. This is very different from the autoimmune diabetes in the NOT model, right? And then when we polyclonal Tregs nearly showed no activity. We think this is because the polyclonal Tregs has alloreactive cells. In fact, when we measure the frequency of alloreactive cells in the polyclonal Tregs, it's as high as 10 to 20%. And so we figured with 20% of the cells reactive alloantigen, if we increase the cell dose by 5-fold, right? And then we will have enough of the antigen-specific Tregs in the polyclonal Treg inoculum to, is it timed? To achieve the same therapeutic effect. That was indeed the case. So if we increase the poly Treg dose to 25 million, now they're very effective in controlling the disease. So whether we need antigen-specific Tregs or not, it really depends on the disease, the condition we are treating. For transplantation, I think we can get away with polyclonal Treg. We just need to give a lot. And the risk of that is perhaps it may be inducing non-specific immunosuppression, not too different than the immunosuppressive drugs we are trying to replace. So how do Tregs work in a transplant setting? And then, so this is the transplant is quite different from autoimmunity because it's a de novo immune response. We know when it starts is when we put in the graft, right? The autoimmune disease, when we treat, is already started. So when we look at this, the Treg treated versus non-treated, here we have the same donor-specific deletion protocol in both groups, so that they're comparable. With Treg therapy, we saw that Tregs prevented the infiltration of the T cells. Without the Treg therapy, you can see this massive CD8 infiltration and with some CD4s. And then with the loss of insulin signal, and then the graft is rejected. This is day 14, before the overt diabetes developed. And then we can see this phenomenon. In fact, if you look at the kinetics of this response, and then this is actually pretty complex slides I will walk you through. If you look at day 4, day 6, and day 14 after transplantation, and then black symbols are those that received donor-specific Tregs, and open symbols are not treated. And then the y-axis is CD8 cell number in the graft, and then the x-axis is CD4 cell number in the graft. Basically, the way we see it is it's a competition of what cells get into the graft first, right? If you don't have Treg cell therapy, and then CD8 actually come in very, very rapidly. And this is before any Tregs can come in. But when you give a Treg cell therapy, the CD8 infiltration is delayed. And in fact, by day 6, you can see actually Treg infiltration is dominating over the CD8 infiltration if you give Treg cell therapy. If you don't give Treg cell therapy, then the CD8 continue to accumulate. And then Tregs, the endogenous Tregs actually don't seem to come in, or insufficient numbers to compete with that. By day 14, though, that the Tregs are in, endogenous Tregs are in, right? In the white open symbol, if you don't treat with Tregs, there are lots of endogenous Treg coming in, but by this time it's too late to control the CD8 response. The graft is already half or mostly rejected. When you treat with the Tregs, and then the Treg remain to be dominant, and then CD4 infiltration never actually can accumulate to large numbers to immediate graft rejection. And so this is in part due to a control of, I don't think I have the slide to show that, it's control of a chemokine expression, and the similar inhibition of a gamma interferon signature, and then CXCL9, CXCL10 expression in the graft to prevent the CD8 from coming in. And if you look at the Tregs in the graft, they if you don't treat it with Treg in early time point, right, they don't come in enough. With Treg therapy is that you allow more Treg to accumulate. Up to 30% the infiltrating CD4s are Tregs. And then this is maintained by the Treg cell therapy. To our surprise, though, that in the first week after Treg cell therapy, half of the infiltrating Tregs have the Li5 marker, are the cells that we inject, and the other half are endogenous Tregs. What's really interesting is that by day 14, our infused Tregs actually in this model did not persist. So it's mostly this Tregs that continue to accumulate or maintain at a high level at day 14. They are actually from endogenous cells. So with therapeutic Tregs, not only they themselves infiltrate the graft, they also help to recruit endogenous Tregs without recruiting the CD8. So that's really interesting. We think this Tregs and then CD8 use different chemokine receptors and to have access to the graft. So in the mice that have long-term graft survival, this is more than 100 days. And then we want to ask, right, since the infused Tregs can no longer be detected after 2 weeks, and then how is the tolerance maintained? And in these mice, if you look in the graph, there's no Tregs in the graft. In fact, there are no CD4s or CD8. It's just a clean graft and no infiltrate whatsoever. And we tend to think at this stage that the graft is actually ignored by the immune system because it's a quiescent state. If you measure the peripheral blood and lymph node lymphoid compartment for donor-specific T cells, they're present in these mice, but they don't bother to go into the islets. And so we have not looked in to see if there are other mechanisms, but the tolerance is, we can be quite certain, is not maintained by Tregs stationed in the graft. So with that, and I'm going to transition into our clinical efforts in both
Can I ask a
autoimmunity.
question?
Sure.
I'm sorry to break in. This is
No
Jane.
problem.
With that last slide, I think that's really interesting that you have this quiescence. Do you know what would happen if there was some kind of perturbation or inflammation in this model? And would you lose that tolerance? Just thinking in humans, how there's so many insults.
Yeah. I want to say this is perhaps unique to islet transplantation. If you have a liver transplantation and kidney transplantation, it could be different. The islet, what's interesting is gradually over time, the islet blood vessel that supply these islets are of host origin. So the islet could possibly hide behind that vascular endothelium, right? And then not be seen by the immune system. But in kidney and liver transplantation, that's not the case. The blood vessel endothelial cells, the vascular endothelial cells are chimeric, are both host and donor origin. And so that could provoke immune response, right? And we actually did the perturbation in these animals by transplanting. I forgot to mention, so thank you for the question. We actually transplant skin graft from the donor mice. And then in majority of the mice, they capped the islet graft, but rejected the skin graft. What we haven't done is to actually cause some kind of infection, right, in the graft, in the kidney, and then see. My suspicion is that they will be rejected. If there's any reason for the T cells to come into the kidney, and the graft will be rejected. Okay. So did that answer your question, Jane?
Yeah, that was really helpful. Thank you.
Okay, thank you. So in the clinical trial, our clinical trial effort, we have coming up, I guess I should just wait until we've launched 11 clinical trials. And so lots of data, they're mostly phase 1, but I want to focus on the pharmacokinetics of therapeutic Tregs, and also our effort in trying to, in phase 1 trials, design the trial to take to have a peek at the efficacy, potential efficacy of Treg cell therapy. So these are the various trials we launched since 2011. Now our program is almost 10 years in operation. And we started our effort in type 1 diabetes. And then it was some lupus and pemphigus autoimmune diseases. And the orange here are in our transplant trials. Right now, we have 3 trials ongoing. In fact, the pemphigus just wrapped up. And then in early 2021, we are about to launch 2 additional trials, one in liver transplantation and one Treg cell therapy in COVID ARDS. So for the data, I'm going to focus on the in terms of the pharmacokinetics, I'm going to focus on the type 1 diabetes Treg trial. This is one we did not get too creative in naming the trial. So this trial is called a Treg and T1D. It was followed up by this trial of TILT, which is Tregs plus IO2. aiming at looking at the pharmacokinetics of the infiltrating or infused Tregs. And then I'm going to present to you our effort on this Artemis trial, which is a liver transplantation. It's a trial that, that, if I have to say, it's my favorite trial because it really is should allow us to take a look at the potential efficacy of Treg cell therapy. And so we have about three different protocols for expanding, for manufacturing therapeutic Tregs. And this is what we call the Gen 1.0 Treg, polyclonal Treg manufacturing. We start with whole blood or leukopak. Then our Treg manufacturing process start with FACS purification as opposed to max purification using three markers, CD4, 25, and 127. And because I think this gives us the highest purity and then also higher yield than MAC purification. And then expansion is just 2 rounds of anti-CD328 bead stimulation with IL-2 feeding in between. And then we are able in 2 weeks time induce about on average 500-fold expansion. So we get about 5 million Tregs to begin with, and then end up with billions of Tregs in 2 weeks. And then these are the trials that use the polyclonal Tregs. So the Tregs actually function better after expansion. They have better suppressive phenotype and a suppressive function. It's shown here is the work actually out of Jane Buckner's lab and up in Seattle is that the type 1 diabetes have a defect in STAT5 in IL-2 signaling, right? In the control subjects, they have higher STAT5 signaling after IL-2 treatment, whereas the type 1 diabetes have this defect. What's interesting is after expansion, you can see that the expanded Tregs in type 1 diabetes subject have corrected that defect. The expanded Tregs also have higher CD25 expression, higher CTLA4, and higher LAP, and they're more suppressive in vitro in this black bar than non-expanded Tregs. And so in terms of looking at the persistence of the Tregs, and then these are autologous Tregs, so it was challenging to determine their pharmacokinetics, but we were able to take advantage of the expertise of our collaborator, Mark Hallerstein, who have developed a protocol to use deuterium to probe immune cell dynamics. And then to do that, we actually cultured expanded Tregs in the presence of a deuterated glucose. It's incorporated into the DNA, now give us a unique marker to the infused Tregs. And deuterium is a stable isotype, non-radioactive, and it allows us to track the cells. Right? Here it shows the deuterium enrichment in the peripheral blood Treg compartment, and the solid lines received the 320 million Tregs, and dotted line received the 2.6 billion Tregs. And you can clearly see the dose response. With more Treg we infused, and we see a higher peak value and a longer persistence of the cells. And what's really surprising to us is that some of the cells that persist all the way for 1 year in the peripheral blood. We can detect them in all the subjects that receive the high dose. In the low dose, and the level actually dropped below the detection level of 0.1%. So this shows us that some of the Treg we infuse, at least some of them can persist, but also shows, and you can see this dramatic drop in the first 3 months. And we don't know what happened to these cells, whether they died and then or they infiltrate the tissue and then are no longer circulating. But one thing we know is that they did not destabilize. because here is the sorting of the cells, right? And then signal from, we can see if the signal is from the Treg population or they're from the conventional population divided into 3 different pots of by CD45RO and CD612. And then you can see here it's 3 months, 6 months, and then 1 year after infusion, we only detect the signal, deuterium signal, in the Treg subset, but not in the conventional T cell subpopulations. And it shows us, at least in circulation, the deuterium containing the infused Tregs maintain their Treg phenotype and then did not destabilize into conventional cells. People always ask this question, so I'm going to show it. It's a phase 1 study, so it's not aimed at achieving any efficacy or be able to detect any efficacy. And we did not see much of a change in C-peptide in the subjects treated in this trial. If you remember from the part 1 of my presentation that we think to treat the organ-specific autoimmunity, such as type 1 diabetes, antigen-specific Tregs, it will have much improved potency. It's likely required to achieve efficacy. But we're interested, I think the Poly Treg therapy trials still have value, right? And then teach us about the potential pharmacokinetics of these cells. And we know that some cells can persist, but that there's this drastic crash in the first 3 months of the cells. And to really answer the question whether this loss of Tregs in the first 3 months is due to the cells disappearing, dying, perhaps from IO2 withdrawal, because they've been cultured in vitro for a long time, for 2 weeks in high dose of IO2. And then going in vivo, if they don't have the sustained source of IO2, they may crash and die. To really address that question, answer that question, we designed this trial we call TILT. And then we combine Treg therapy with low dose IO2. And then here's the trial design. We infuse Treg on day 0. And then in the first week after Treg infusion, starting on day 3 to day 7, and the subjects received 5 subcutaneous injection of IO2. And it's the IO2 dose is at 1 million unit per day per injection. And later on, we actually have to decrease because we saw some subjects are losing C-peptide and decrease the dose to 0.3 million per day. And then this, the IO2 dose is repeated in some of the subjects from day 38 to day 42. And remember, this is when we actually saw the drop. This is after the drop, right? And we're hoping that this will help to sustain it. Perhaps the repeated IO2 is needed to keep the level of the infused Tregs needed. So we designed 2 doses of IO2 in the TILT study. And so what's the impact of the IO2 on top of the Tregs? So if you look at the low dose IO2 clearly worked. If you look at the just total Treg percentage in the blood, and then these gray bars are the period of IO2 treatment, you see is at the end of the IO2 treatment, you see a sharp rise of percentage of Tregs in the peripheral blood. This is total blood, not the total Tregs, not infused Treg. And then it's followed by a drop back down to baseline. And if you treat it again, they rise again and then drop again. And then by looking at the deuterium, we can look at see the impact of the treatment on the infused Tregs. And then here I overlaid in gray, the solid field, are the type 1 diabetes, our first Treg trial, right, without the IO2, and then the pharmacokinetics of the Tregs. These are the 3 subjects, I think, received the similar dose of the Tregs to the subjects in the TILT study. And you can see clearly, I hope, that there's a benefit of the IO2, low dose IO2 therapy in sustaining the infused Tregs. And what's interesting is the effect is actually quite long lasting. The treatment stopped at 42 days. The second treatment is 42 days is right here, right? You can see the benefit at 3 months, but it persisted all the way to 1 year. So we think, the Treg low-dose IL-2 therapy will be indeed beneficial, and then likely the decline we saw in our first study is due to IL-2 deprivation to these Tregs.
Tang, can you distinguish between the subjects that got the second IL-2 dose on the, I can't tell the color.
Yeah, I can. Sorry, I just, for simplicity, I took that away. In fact, I think, for example, this orange subject and also the brown did not receive the second dose. You don't see the rise, right? And there was a pretty close correlate with the dose of the IL-2 and also the whether they received the second dose or not. On here, I cannot be sure that the same color was used though. I have to go back, and then the legend was so big because it's 2 different doses and different cell doses, and I removed the legend for simplicity. I want to say that the black and the red here did not receive the second dose of IL-2.
Thanks.
Yeah. And it's not only just increasing the number of Tregs, and then if by single cell RNA-seq, when we look at the Tregs, total Treg population, right? Not the infused Tregs. In the TOAD study here, we also have the banked sample from our first trial. And then you can see here, this little triangle is actually FOXP3 positive cells in Treg population. By looking at their gene expression, we can see that the IL-2 therapy also enhanced the Treg markers, right? The FOXP3 expression, and then these are the peaks. It's right at the end of the IL-2 treatment, GARP, GITR, and then CD27, and then also HLA-DR, I believe. So these are all IL-2 responsive genes in the Tregs that responded to the IL-2 treatment. And we don't see that change of that signal in the Treg T1D trial. So in addition to its effect on Tregs, the low-dose IL-2 therapy also increased, give us this quite dramatic granzyme signal, a cytotoxic signal, both in NK cells and also in CD8 cells. And then here is in the, so here, David, I can tell you that this is the subject, the blue line, received 1 million Tregs only in the first dose, because this subject showed a reduced C-peptide by day 28 and then did not receive the second dose of the IL-2. So it has the highest rise. This green subject also received the 1 million IL-2. Did I say 1 million Treg? So both of these subjects, the top 2 subjects, received the 1 million IL-2. And then the rest of them received the 0.33 million. So there is this dose effect in terms of the granzyme B response to the low-dose IL-2. We don't think this is due to the Treg therapy, because we really don't see the response, a granzyme B in NKs or CD8s in our Treg type 1 diabetes study. In fact, the granzyme B positive CD8 is a signature population in this patient subject. And here is a single cell RNA-seq and combined with TCR sequencing. And then the dominant TCR clones mapped on top of the UMAP, right, of a different subsets of cells in the peripheral blood. Here I highlight the NK cells is here, is this, I somehow see a mouse or animal, but this is a head of the animal. right? And here is the granzyme B positive CD8 is where these cells are concentrated. And then this little protrusion here, the population 6 is MATE cells, mucosal associated invariant T cells, and it's over here. What's interesting is in type 1 diabetes Treg therapy, you see a dominant population of these granzyme B positive effector CD8s. And then this population is also present in IL-2 treated subjects, but we see a emergence of a new subset. It's the MATE cells, the mucosal associated innate immune cells, and suggesting that these cells may be responding to IL-2 treatment. And then this is their effect on type 1 diabetes, on autoimmunity, it's not completely clear, but it could be potentially pathogenic.
Do you think the CD8 response is also increased? It looks like it is.
The CD8 responses by flow cytometry, we could confirm that. Yeah, the CD8 over time, it was CD8 increased over time with IL-2 treatment. Okay, so with that, that's all I'm going to show for the polyclonal Treg cell therapy. But I want to now switch gear to donor-specific Treg therapy, and then we have an opportunity to do this in transplant settings because the precursor frequency is really high. They're readily expanded in vitro, in short-term in vitro cultures. The way we do it is the same Treg purification, but we replace the first round of anti-CD328 bead stimulation with activated B cells. And then these B cells are from donor. They've been previously activated with CD40 ligand expressing K562 cells, in addition with to IL-4. So we expand these B cells and activate them, and then they become potent stimulator of Treg expansion. So in about 7 days, and the Treg that remained in the culture are all donor specific. We then allow the cells to rest. By day 11, we re-stimulate with anti-CD328 beads to expand the cells more, to increase cell number, and then by day 16, we formulate and infuse into patients if it pass release. So then here is showing the expansion kinetics. Here's using healthy donors with mismatched DR. And then after expansion, they remain to be TSDR demethylated. And then they have a strong suppressive activity in vitro when you measured against the alloantigen used for the expansion, but not with third party. Here is not with third party. This is showing the gain of suppressive activity compared to polyclonal Tregs. And then with this Treg cell product, and then here are 5 transplant trials we launched. And I'm going to focus on the Artemis study. And in the one doing designing transplant studies with Treg cell therapy, and we have many choices, right? And there are many organs transplanted, and most of the organ transplanted is kidney, and then followed by liver, but heart and the lung and the intestine need more help because they don't, the graft function are not as robust as the kidney and the liver. But really in these early trials, and then we want to do no harm. And also I really would like to see a signal to encourage further development. And then in the end, we think the liver is the best place to do this because they themselves are tolerogenic and then require lower immunosuppression. You can potentially take the drug off if the patient rejects, and you can treat that rejection with no long-term consequence. And that cannot be said with any of the other transplants. Another interesting thing is that in our lab, we've worked on mouse liver transplantation. Mouse liver transplantation is spontaneously tolerated. So they go through this burst of proliferation. Alloreactive cells will proliferate to the transplanted liver, but eventually they are suppressed, and then the alloimmune response is suppressed. And here is showing here that if you measure anti-donor response in the naive animal, you can readily detect donor-specific CD8, CD4s. And then this is not donor-specific CD8, but it's Tregs, but the total Treg population is of a typical naive animal. But in liver transplanted mouse that already tolerated the liver, right? And then you see a dramatic deletion of donor-specific CD8 cells, reduction of donor-specific CD4 cells. If you look at these CD4 cells, they're mostly FOXP3 positive. Not mostly, they're 30 to 50% FOXP3 positive. So they're suppressed, right? So I think the liver is already doing half of the job, but in humans, they're not spontaneously tolerant, so they could use help. So it's an ideal situation to design a clinical trial to allow us to test experimental drug withdrawal to see if we can see a signal. In fact, experimental drug withdrawal is already happening in the clinic in patients. And this shows that if you take patients 2 to 6 years after liver transplantation and gradually reduce immunosuppression, and then you can get away with about 50% reduction of immunosuppression until patients start to experience rejection, right? As you reduce more, and then the more would reject, ultimately about 15% of the subject, actually 2 to 6 years after liver transplantation, will achieve tolerance. And they will not reject. In the absence of immunosuppression, will maintain immunosuppression free for more than a year. That's our definition of tolerance, right? This gives us a platform to test tolerance promoting therapy, such as Tregs. So what we decided to do is to infuse the donor-specific Tregs we expanded for the patient right before the 50% reduction of immunosuppression. And then we ask whether we can change this curve of rejection. And our primary endpoint is the percent of patients who can come off immunosuppression, reduce immunosuppression by 75%. And our secondary endpoint is a patient who can be tolerant and then come off immunosuppression for 1 year. And so these are adult liver transplant patients and with living donor, because we need a donor material to manufacture the alloreactive Tregs, the donor reactive Tregs. There's 2 to 6 years after transplantation. When we did a power calculation, we actually think we only need 9 subjects to be able to detect the signal, whether the Tregs can change this rejection curve or not. So we aim to enroll 11 subjects and infuse 9 subjects. And then we specified that we want to infuse at least 300 million of donor reactive Tregs to permit 100% immunosuppression withdrawal. And we also specified that we want to infuse at least 100 million Tregs, donor-specific Tregs. If we cannot manufacture for some reason 100 million Tregs, we will not infuse, and the patient will have to come off the study protocol. But if we infuse between 100 million and 300 million, the patient can only come off immunosuppression to 75%. And then this, by our clinician, Sandy Feng, who designed the trial, think this is the safest for her patients. And then the study enrolled patients at UCSF, Mayo Clinic, and then Northwestern. So I'm going to go through, the trial has completed, and we're writing the manuscript and finalize the mechanistic study right now, and I'll walk you through the clinical results. So this is the first subject we enrolled, and then across on top is the stage of immunosuppression withdrawal. And then the time of AR-Treg infusion, continued immunosuppression withdrawal, primary endpoint, and then secondary endpoint. So this first subject we enrolled, we end up manufactured 189 million Tregs, not reaching the target dose of 300 million. And this subject also decided to go home and decided that she's going to stop all her immunosuppression altogether. So she went home and then after the infusion stopped all her immunosuppressive drugs. And about 8 weeks later, she experienced a T-cell mediated rejection and came off the study. So it was not terribly informative. So the next 3 subjects, and 2 of them, we manufactured a target dose of Tregs, and we infused 378 million and 461 million in the next 2 subjects. And both of them actually got to the primary endpoint. What's interesting is that the second subject at the time of primary endpoint assessment were found to have abnormal liver test, and then was declared to have a T-cell mediated rejection by pathology, a mild T-cell mediated rejection by pathology. So he officially failed the primary endpoint, whereas the third subject met the primary endpoint. But the subject actually declined to continue immunosuppression withdrawal, so we were not able to assess the secondary endpoint. The fourth subject, we only manufactured 136 million Tregs, and then the patient also at the time of scheduled Treg infusion had diarrhea, a GI symptom, but nonetheless, the physician deemed it's okay to infuse the Tregs, so infuse the Tregs, and shortly after, I think 10 days or 2 weeks, and the subject have a rise liver enzyme, and then pathology showed that it may have T-cell mediated rejection. So the patient came off the protocol. And the next 3 subjects were not able to manufacture 100 million Tregs, so that the infusion did not happen. The next subject, we produced 126 million, meet the 100 million infusable dose, and then the patient reached the primary endpoint, but because she did not receive the target dose, and then she actually stopped immunosuppression withdrawal and maintained at its 25% original level. And the next 2 subjects, one actually had a rejection before Treg manufacturing, and another one, the Treg manufacturer did not meet the dose. So this is the entirety of the trial over a course of, I think, 2.5 years. So as you can see, we have a major problem in manufacturing the Treg. Although we designed the study to be powered to detect an efficacy, we couldn't actually infuse sufficient number of patients and sufficient dose to be able to assess efficacy in the study. So manufacturing was a big problem. My role of this whole entire trial was overseeing manufacturing, so the pressure was extremely high. And so we're trying to understand what's the problem. And then here showing you the individual patient that we manufactured, the Treg expansion in the culture, right? These are the 2 subjects who expanded well, reached the target dose, and then received the target dose. And then these are the subject received partial dose, and then those are the ones that we did not even meet the minimal infusible dose. And for one subject, the brown line here, we tried twice, and then the donor the patient was very committed and then want to give blood again. And the second time, we leukapheresis the patient and then used more Tregs to manufacture, and still could not meet the target, the minimal infusible dose. This problem seems to be very specific for Artemis. So here's all the AR alloreactive Treg we manufactured in the similar period. And this is the Artemis patient we manufactured their fold expansion versus non-Artemis subjects, right? You can see a statistically significant less expansion. So this got us to think what's different from the Artemis patients than these. These patients are liver transplant patients or pre-transplant patients. And then one major difference is that the Artemis patient have a donor liver in their body, right? You know, back in graduate school days, when we were told that the livers is the graveyard of T cells. So the livers can kill T cells, antigen-specific T cells, and also where T cells go there to die. So is it possible that having a donor liver in the body, that the Tregs are either deleted or at least trapped inside the liver? They're not in the peripheral blood. It's what we use to manufacture these cells. So we decided to perform a analysis of the frequency of donor-specific Tregs and of Artemis subjects, and then versus pre-liver transplantation and then pre-kidney and pre-lung transplant patients. And these are all samples collected at UCSF. And then as you can see here in these Artemis subjects that already received their liver transplantation, the frequency of donor-specific Tregs is much lower. So the reason we cannot manufacture Tregs for Artemis is because these cells are actually not in the peripheral blood because they have received the donor liver allograft, and then they are trapped in the liver or deleted by the liver over the course of 2 to 3 years after liver transplantation. Okay, so I didn't bring this. So for this pre-liver transplantation is not Artemis subjects. It's another cohort of patients. We have now confirmed this result with a serially collected sample of liver transplantation from pre-transplantation to 2 years after transplantation to show that actually this loss of Tregs in the peripheral blood, donor-specific Tregs in the peripheral blood, it occurs quite rapidly, as early as 6 months after transplantation, and it's persistent to 2 years after transplantation. It's not just donor-specific Tregs, it also affects donor-specific CD8s and then conventional T cells. So here's a summary of this clinical part of it, and we have conducted many clinical trials and then infused hundreds of patients, right, up to 2.6 billion. The hundreds of patients is collectively in the field, not only at UCSF. I think we have maybe 50 patients. So far, all reports that agree that the Treg therapy has been well tolerated, and then some of the infused Treg can persist for a long time and then maintain the Treg lineage and the stability. And you can potentially use low-dose IL-2 to improve Treg persistence and therapeutic Treg persistence. And in some settings, we have seen favorable immunological impact of the Treg therapy, right? But there are lots of challenges. And then, first of all, I have just shown you that antigen-specific Tregs may be not in the peripheral blood, so that making manufacturing of antigen-specific Tregs challenging. In some patients, Treg may be defective. I showed that the Treg expansion can correct some of the defect, but it's not a permanent, may not be a permanent correction, or there are certain defects that may not be corrected at all. Manufacturing Tregs for autologous therapy has been highly variable, highly challenging. I think the problem tend to be with patients who have systemic inflammation, and then we had some challenges with lupus patients as well. And then so we think that the immunological profile of the subject will greatly influence how well the Treg would expand. So we need more robust protocol. Low-dose IL-2 can sustain the infused Tregs, but it's not Treg specific, so we need a better solution to that. And then also to generate antigen-specific Treg, autoreactive Treg, for example, has not been feasible. And yet we, being a live drug, and even though we haven't seen Treg destabilization in subjects in the peripheral blood, and this is always a concern, how could Treg destabilize and then cause harm? So with that, I think I'm actually over my time. I was going to go to the third part of the talk on next generation Tregs.
So
should I continue?
So we are
Maybe not.
We are over time.
Sorry about that. I did not see my clock.
So why don't we open it up for some questions? So that was amazing. Thank you so much for that talk. And we'll have to have you back for this next part. But a lot of your talk, even the human data on persistence, showed long-term circulation, out to a year. And then all of your donor-specific T-cell work in the mouse was specific at the islet. So how big of a role is circulation, and does the draining lymph node, is that necessary? Have you ever tried to block infusion or block migration to the lymph node to see if that's necessary?
So in the NOD model, we know we think they directly go into the islets. They are chronic inflammation. In the NOD mouse, it's a specific feature of the NOD mouse is that they actually set up tertiary lymphoid organs that can even directly recruit naive cells. So we see infiltrating Tregs infiltrating the islet overnight after injection. So I think they directly go into the islet. We have not done the experiment. It will be difficult to prove, but I don't think they need to go into the lymph node to suppress the immune response in the islets.
So if they get to the tissue, you think it's sufficient?
I think so. In fact, we in the long term, 1 to 2 months after the Treg infusion in the NOD mouse model, you can hardly find any Tregs in the lymph node or the spleen, and let alone blood. And they're mostly very few of them, less than 10 per islet in the pancreatic islets.
Other questions? I didn't see anything in the chat box.
Well, I have a question about the Artemis experiments. Very provocative. Of course, liver transplants are being done at different ages for different indications. Did you try to make sense based on the age of the donor, on the reason for the transplant? Was it secondary to hepatitis C, or was it alcoholic induced hepatic failure? Was there any sense to that heterogeneity of the donor and the recipient?
Right. So the study is in adult. It definitely had a wide age range. I think our youngest subject was 23. Our oldest subject was 70. Among the patient, we manufactured Tregs, and then the older ones are harder to expand Tregs for them. In terms of the liver transplantation tolerance and the immunosuppression withdrawal, and definitely pediatric patient population, you can achieve higher percentage of successful withdrawal than adult population. In our trial, we have excluded autoimmune indications. We originally, when we designed it, this is how long ago it was, that we designed to exclude hepatitis C. That's because then they could have recurrent hepatitis C, they could have infection that was just more complicated. But then in the process of running our trial, getting regulatory approval and all of that, and Hep C drug become available. So we actually allow the patient successfully treated with Hep C to enter our trial.
Yeah.
So we have Hep B, Hep C, and alcoholic liver disease, and all included in the study. But I don't quite remember the specific breakdown of these subjects we have.
Thank you.
Mhm.
Other questions?
I have a question on in the organ transplant setting, Tang. Your mechanistic studies in the islets are beautiful. I was wondering how much you've been able to look at what the key regulatory function is in the liver transplant model. Is it similar that you're modulating CD8 responses primarily, or?
So in the mouse model you're talking about, right?
Yeah.
Yeah, in the mouse model, in the liver transplantation is spontaneously tolerant. You don't have to treat with Tregs. What happens is that the CD8s are deleted. The liver themselves deleted the CD8s. The liver sustained massive damage. I think in patient we'll call that rejection. But the liver regenerated once the CD8s are lost. And then they regenerate and then mouse were okay. But this happens over the course of 1 month. And then CD4 T cells there was some CD4 deletion, donor-specific CD4 deletion, but a lot of them actually acquired a Treg phenotype. We don't know whether it's because of preferential, we haven't done the mechanistic study to distinguish whether it was a selective deletion or preferential deletion of effector cells, a retention of regulatory T cell, or de novo induction of regulatory T cells. But there's CD4 that remained is high, has a high percentage of FOXP3 positive cells. If you challenge these mice with a donor graft, skin graft, the graft is not rejected. The skin remains, so very different from the islet transplant model. But underneath the skin is packed full of Tregs.
Interesting. And is there have you worked in another organ transplant model where you can look at the mechanistic role of the Tregs?
Right. So in the islet model, we did look, and it's controlling of the infiltration and preventing the dominance of the CD8. And also the few CD8 that got in actually expressed PD1. This is before the PD1 was so hot for cancer immunotherapy. So we think it's not only preventing them to come in and also inducing the CD8 dysfunction.
I have a question.
Yeah.
Okay. So I was just wondering, because you emphasize about the infectious tolerance. So in some of your studies, have you actually looked at if the endogenous Tregs are actually playing a bigger role at a later time point compared to because your exogenously added Tregs apparently persist for a very long time, right? So
In the NOD model, yes. In the NOD mouse model, it persisted, whereas in the B6 islet transplantation model, they did not. They last about 2 weeks, and that's the last time we could detect some of these transfer Tregs. We don't know why that is. So in that model, in the islet transplantation model, I think endogenous Tregs ultimately took over.
right.
To control the rejection, I guess, is through this infectious mechanism.
Right. So are they as potent as the adoptive transfer Tregs? They're antigen specific, right? I would assume. That's
right. We have not done that experiment. But if you isolate the Tregs out of the peripheral lymphoid organs, and I think that it will not be all antigen specific, right? So that will be a harder experiment to do. You have to have a way of tetramer or whatever to be able to pull out the antigen specific cells and then quantify them. First of all, do they expand? Once you use Tregs to induce tolerance, do you expand donor specific Tregs? And then to be able to isolate them to show that if they function. We have not done that, but Herman Waltman at Oxford have done those kind of studies in transplant models, and to show that you can actually serially transfer tolerance. And then he coined the concept of infectious tolerance.
Thank you.
Other questions for Tang? Well, I'd like to greatly thank you for being rescheduled and giving us this amazing talk. I think we can give everybody a break here before your next session, but thanks again for joining us.
All right, great. Thank you
Thank you.
so much.
Great.
All right. Okay, bye-bye. Let me see how I'm gonna stop the sharing. There we go.